Cellular respiration is a set of catabolic reactions that converts chemical energy from nutrients into adenosine triphosphate or ATP in the presence of oxygen. Nutrients that are commonly used by animal cells in respiration include carbohydrates, amino acids, and fatty acids. In this video, I'll summarize how these macromolecules are oxidized to produce ATP during cellular respiration. Cellular respiration for carbohydrates starts with glycolysis, which is a 10-step process that breaks down the 6-carbon glucose into 2 molecules of the 3-carbon pyruvate. Glycolysis occurs in the cytosol and generates a net total of 2 ATP molecules through substrate-level phosphorylation, and 2 molecules of NADH, which stores potential energy that can later be harvested in the electron transport chain. As I covered in my previous video on glycolysis feeder pathways, other sugar molecules enter glycolysis through different intermediates of the pathway. Next, pyruvate translocase co-transports pyruvate and proton into the mitochondrial matrix, where pyruvate undergoes oxidative decarboxylation by pyruvate dehydrogenase complex to generate acetyl-CoA. This reaction also generates a molecule of NADH. Acetyl-CoA then enters the citric acid cycle, an 8-step process that generates a net total of 2 carbon dioxide, 3 NADH, 1 FADH2, and 1 GTP per cycle. Note that the 6th step of the citric acid cycle, succinate dehydrogenase, is also the second complex of the electron transport chain. The final step of cellular respiration is oxidative phosphorylation, which involves the flow of electrons through a series of complexes located on the inner mitochondrial membrane, which conserves the free energy of fuel oxidation as a transmembrane electrochemical potential that provides the free energy of ATP synthesis through chemiosmosis. The first complex, known as NADH dehydrogenase, oxidizes NADH and reduces ubiquinone, abbreviated as Q, through a series of redox reactions. The energy of this favorable electron flow is stored as electrochemical potential by pumping 4 protons from the mitochondrial matrix to the intermembrane space. The second complex, succinate dehydrogenase, which is also the 6th enzyme of the citric acid cycle, oxidizes succinate and also contributes to a pool of reduced ubiquinone through a series of redox reactions. Note that the second complex doesn't pump protons out of the mitochondrial matrix. The third complex, ubiquinone cytochrome C oxidoreductase, reoxidizes ubiquinone and reduces cytochrome C through a series of redox reactions, while pumping 4 protons out of the mitochondrial matrix. The fourth complex, cytochrome C oxidase, oxidizes cytochrome C and reduces oxygen to water. Therefore, the purpose of oxygen during cellular respiration is to serve as the final electron acceptor of the electron transport chain. Complex 4 pumps an additional 2 protons out of the mitochondrial matrix. All the protons pumped into the intermembrane space creates an electrochemical gradient known as the proton motive force. During chemiosmosis, protons flow down their electrochemical gradient through ATP synthase, causing a conformational change that converts ADP and inorganic phosphate into ATP, a high-energy compound that drives many anabolic reactions in our body. Phosphate translocase on the inner mitochondrial membrane co-transports protons and inorganic phosphate into the mitochondrial matrix, while adenine nucleotide translocase transports ADP into the matrix and ATP out of the matrix. The 2 NADH molecules produced during glycolysis need to find a way to enter oxidative phosphorylation, since the inner mitochondrial membrane is impermeable to NADH. In the liver, kidney, and heart, the malate aspartate shuttle utilizes cytosolic and mitochondrial malate dehydrogenase to transport NADH across the inner mitochondrial membrane. The NADH molecule then enters complex 1, generating a net total of 32 ATP per glucose molecule. On the other hand, in skeletal muscle and brain, the glycerol 3-phosphate shuttle utilizes cytosolic and mitochondrial glycerol 3-phosphate dehydrogenase to transport NADH, which is used to reduce FAD and subsequently coenzyme Q. As a result, the glycerol 3-phosphate shuttle only generates a net total of 30 ATP per glucose molecule. Glycolysis, citric acid cycle, and oxidative phosphorylation constitute the central catabolic pathway in our body. Amino acids may enter these pathways through different intermediates. Glucogenic amino acids can be degraded to pyruvate or intermediates of the citric acid cycle, including alpha-ketoglutarate, succinyl-CoA, fumarate, and oxaloacetate. Ketogenic amino acids can be degraded to acetyl-CoA or acetoacetate, which is converted to acetoacetyl-CoA by beta-ketoacyl transferase, with succinyl-CoA as the donor of coenzyme A. Acetoacetyl-CoA can then be cleaved into 2 molecules of acetyl-CoA by thiolase. Fatty acid catabolism starts in the cytosol, where fatty acyl-CoA synthetase condenses fatty acid with coenzyme A to synthesize fatty acyl-CoA, which is coupled to the hydrolysis of ATP into AMP and inorganic pyrophosphate. The inner mitochondrial membrane is also impermeable to fatty acyl-CoA. Therefore, fatty acyl-CoA enters the mitochondrial matrix through the carnitine shuttle. Once fatty acyl-CoA enters the mitochondrial matrix, it undergoes repeating 4 steps of beta oxidation, which cleaves off 2 carbons from the carboxyl terminus as acetyl-CoA. Acetyl-CoA enters directly into the citric acid cycle. The first step of beta oxidation involves acyl-CoA dehydrogenase, which oxidizes fatty acyl-CoA into enoyl-CoA, while reducing FAD to FADH2. The FADH2 then transfers electrons to electron-transferring flavoprotein, abbreviated as ETF, which in turn passes electrons to the ETF ubiquinone oxidoreductase, ultimately reducing ubiquinone. In the third step of beta oxidation, beta-hydroxyacyl-CoA dehydrogenase oxidizes beta-hydroxyacyl-CoA into beta-keto-acyl-CoA, reducing NAD+ to NADH. The NADH directly enters complex 1 of the electron transport chain. In summary, our body oxidizes carbohydrates, amino acids, and fatty acids in various catabolic pathways, storing chemical energies as reducing equivalents, NADH and FADH2, which can then be harvested by the electron transport chain as proton motive force to generate ATP through chemiosmosis.